This is Chapter 7, Cellular Respiration. Cellular respiration, or aerobic respiration, because it involves oxygen, is the opposite of photosynthesis. We're going to take a glucose molecule, that's C6H12O6, add some oxygen to it, and then break it down to create carbon dioxide, which will be a waste gas, water, which will be used in other places in the cell, and the most important part, ATP, that's cellular energy. This is going to happen in 4 separate steps: glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation or chemiosmosis. And I will talk about each one of those one by one. We'll start with glycolysis. Glycolysis is the splitting of glucose. It takes place in the cytoplasm. Glycolysis is the result of a biochemical pathway. We'll begin with the 6-carbon sugar glucose. We'll add 2 ATP molecules to that. The energy from the ATP molecules will split glucose into 2 3-carbon sugars. In addition, the phosphate from those ATPs will be added to those 3-carbon sugars. This process is called phosphorylation. As each of these 3-carbon sugars enter their own biochemical pathway, 3 things will happen. The 3-carbon sugar will be modified and turned into pyruvate or pyruvic acid. The coenzyme NAD will have a hydrogen ion added to it and become NADH, which is an electron carrier. And the last thing is 4 ATP molecules will be made. So at the end, we have 2 pyruvic acid molecules, 4 ATP molecules, and 2 NADH electron carriers. Remember, we started this process with 2 ATPs, so we only have 2 extras at the end of this. This slide is just to point out where the ATPs went in and where the ATPs came out in this process. Even if you don't remember the exact steps in glycolysis, these are the things you need to know. It occurs in the cytoplasm. 2 ATPs were produced because we used 2 ATPs at the beginning. 2 pyruvic acids were made, and 2 NADH electron carriers were formed, and these we'll use later for ATP creation. Before we continue with the idea of cellular respiration, we have to look at the mitochondria. The outer membrane you already know, that's that separation between the organelle and the cytosol. The inner membrane is important because it is the location of both ATP synthase, the enzyme that is going to be used to create ATPs during oxidative phosphorylation, and it is also the location of the electron transport chain. The crista, or the cristae, if you're talking about more than one, are the folds of that inner membrane. These folds increase the inner membrane surface area to increase the number of ATPs that can be made. And finally, we're looking at the matrix. The matrix is the space inside the inner membrane. Hydrogen ions will be pushed across this to produce ATPs, NADH, and FADH during the Krebs cycle. The next step in cellular respiration is pyruvate oxidation. Oxygen must be present in order for this to happen. If there is no oxygen present, this step is changed to one we'll talk about later. When the oxygen is present, the pyruvate moves into the mitochondria, specifically to the inner membrane. The pyruvate or pyruvic acid is modified in 2 steps. First, oxygen is added, and then a coenzyme is attached. This turns the molecules into acetyl-CoA molecules. These are 2-carbon molecules. In addition, carbon dioxide is created, and this will be released as a waste product. 2 more NADH electron carriers will be created when 2 hydrogen ions are added to 2 NAD coenzymes. The total energy created for this process will be those 2 NADH molecules. Our next step is the Krebs cycle, also known as the citric acid cycle. Each of the acetyl-CoA molecules will enter the Krebs cycle alone. The carbons in the acetyl-CoA will each be converted into CO2. The acetyl-CoA combines with a 4-carbon molecule called oxaloacetate to form citric acid or citrate. Citrate is a 6-carbon molecule. As the citrate moves through the Krebs cycle, it will be converted into several different molecules. The first of these reactions will each release a carbon in the form of carbon dioxide. The final molecule formed is oxaloacetate. This allows the cycle to begin again. For each turn of the Krebs cycle, 2 carbon dioxide molecules are created, 1 ATP is formed, 3 NADH molecules are formed, and 1 FADH molecule is formed. This is where we are in terms of energy made. So far we have 4 ATP, 2 that were made in glycolysis and 2 that were made in the Krebs cycle. And we have a lot of electron carriers. 2 NADH made in glycolysis, 2 made in pyruvate oxidation, 6 made in the Krebs cycle, and 2 FADH that were also made in the Krebs cycle. We'll be moving on to chemiosmosis to change all of those electron carriers into ATPs. Our next step is the electron transport chain. We're going to begin to change those electron carriers into actual energy. The NADH and FADH will move the electrons to the electron transport chain. Hydrogen atoms will be split into hydrogen ions and electrons. The electrons will move down the transport chain, releasing their energy, just like they did in photosynthesis, until they get to the final electron acceptor, which is oxygen. There, some of the hydrogens will combine with that electron acceptor and create water. The other hydrogens will move on to the chemiosmosis step. The last step is chemiosmosis. Chemiosmosis is the movement of ions across a selectively permeable membrane, creating energy in the form of ATP. It's going to occur in the inner membrane space, and the matrix. It's also going to occur at the same time that electrons are being passed down the electron transport chain. This is how it works. The hydrogen ions that were split from their electrons but not turned into water will be pumped across the inner membrane of the mitochondria into the inner membrane space. This will create a concentration gradient, just like it did in photosynthesis. The hydrogen ions will diffuse through the membrane by way of ATP synthase. They will move into the matrix where ADP molecules and individual phosphates will be waiting. The hydrogen ions will combine the ADP and the phosphates into ATP. This process is called oxidative phosphorylation. So now it's time to do some math. We have 2 ATP molecules that were created during glycolysis and 2 that were made during the Krebs cycle. We've now converted the NADH and the FADH molecules into ATP. NADH molecule is the same as 3 ATP, and the FADH molecule is the same as 2. So when you total everything up, you get 38 ATP created. Prokaryotes carry out cellular respiration slightly differently than eukaryotes. This slide shows the ATP yield for a prokaryote. If we were looking at eukaryotes, our total ATP yield would be 36. But either way, these are theoretical yields. Living organisms are not as efficient as the math would have on the paper. A eukaryote will actually only create 30 ATP per glucose, and this is due to two reasons. One, the inner membrane is leaky. Some protons are able to move across the membrane without moving through the ATP synthase, so they don't make ATP. And second, the eukaryote cells actually steal from themselves, and some of those hydrogens in that hydrogen gradient are actually used for other purposes than ATP. When you combine those two together, that's where those 6 ATPs go to. There are some cells that can't use oxygen as the final electron receptor in cellular respiration. Those cells have to rely completely on glycolysis to create ATP, since glycolysis is the only part of this process that is anaerobic. Remember, anaerobic means that you use something other than oxygen. Since they can't carry out any of the processes that require oxygen, they can only make 2 ATPs. This wouldn't be enough energy for the cell, so they continue on, but their process will be different, and it will be called fermentation. There are two types of fermentation: ethanol fermentation and lactic acid fermentation. Ethanol fermentation is used to make bread rise because one of the waste products is carbon dioxide. It's also used to make beer and wine because a second waste product is ethanol, the alcohol in beer and wine. Lactic acid fermentation occurs in animal cells, particularly the muscle cells, and it occurs as the result of exercise. As I discuss these, I will flip back and forth between this slide and the previous slide. When we talk about ethanol fermentation, we're going to go back to the top. We're going to start with glycolysis and turn glucose into pyruvic acid. When we do that, we'll make those 2 ATPs that we talked about. Those pyruvic acid molecules will be converted into acetaldehyde. When that happens, the carbon dioxide as a waste product will be created. The acetaldehyde will then be transformed into the ethanol. But what's more important is the NADH that is created. Remember, each NADH molecule is the equivalent of 3 ATP. So 2 NADH molecules will create another 6 ATPs for the cell. If we're talking about lactic acid fermentation, we're going to go back to the top. We're going to start with glycolysis, and we're going to turn that glucose molecule into 2 pyruvic acid molecules. And again, we're going to create 2 ATPs. Then we're going to turn those pyruvic acid molecules through a series of smaller reactions into lactic acid. That lactic acid then will produce those extra NADHs that we talked about in ethanol, creating those 6 ATPs. You experience the creation of lactic acid anytime you do a large amount of exercise. When you exercise, your muscles require a lot of energy. To do this, they convert enormous amounts of glucose into ATP, which requires a lot of oxygen. If you continue to exercise, your muscles can't keep up with the demand, meaning they can't convert with that oxygen, so they have to switch to anaerobic respiration, and that creates lactic acid. The lactic acid will begin to build up. Circulating blood will remove that excess lactic acid. But if you stop exercising suddenly and you don't stretch out, that lactic acid will build up. It won't be removed quickly, and you will experience fatigue and cramping. This is the end of the cellular respiration chapter.